Good morning. My name is Dmitry Matyushev. I am from the Department of Physics and School of Molecular Sciences at ASU. I am also affiliated with the Center for Biological Physics. Today's lecture is about feeding biology with electrons. So what I'm going to talk about is how energy is produced in biological systems. So the main topic of this lecture is really bioenergy. So the questions which we are trying to address here is how energy is produced in biology, how efficient it is, what are the sources of energy, and where it eventually goes to. So before we start, it's useful to discuss what are the scales of energy and what are the dimensions of biological systems. So the typical scale of energy in biology is 1 electron volt. So this is the energy which is required to move electron through 1 volt of potential in between plus and minus in a battery. So if you switch the units, you can represent 1 electron volts as about 23 kcal per mole. So kcal is kilocalorie. This is the same as capital calories that you usually see on food packaging. So 23 capital calories is something probably close to half of an apple, but we need to divide this energy by 1 mole. And 1 mole is a very large number. This is 10 to the 23 particles, which means that if you divide this half of an apple by this very large number, which would be billions of particles, then of course the energy is very small. However, this is the energy which we see on the molecular scale, because molecules of course are small. At the same time, this energy is what we observe for light. So for instance, 1 photon of visible energy is about also 1.5 electron volts. So the photon here is the smallest amount of energy the light can possibly have. And of course, in a battery, you drive billions and billions of electrons between 1 volt of voltage, but each electron carries the same energy of 1 electron volt. That's why the potential of the battery is about 1 volt, typically for a lithium battery usually used. So the length scale of the molecules and complexes that we are talking about is we need to start from an atom again. So atom would be something like of a few angstroms, and 1 angstrom is 10 to the negative 8 of a centimeter. So it means in 1 centimeter you can put 100 million atoms. So the biology uses proteins and protein complexes to produce energy, and those are of course bigger molecules made of atoms, and the typical size is already in nanometers. Nanometer is 10 to the negative 9 of a meter. So the typical protein complex will be between 20 and 100 nanometers, which would be about 200 to 1000 angstroms. So of course these are larger systems, but not too large, so they're still on the so-called molecular scale, which means we can still measure them in angstroms. We do not have to use other units like centimeters to understand the scale of biological systems. And so of course those molecules are still too small to see them, but you can use different techniques, for instance X-ray spectroscopy, to resolve those structures. And this is a typical picture of how a protein would look like in a membrane. So here is a membrane, which is essentially the walls of the cell, and you can see that the gray area are lipids and the red balls are phosphates, and in the middle is this a protein complex made of two identical proteins, which is inserted in the membrane. So this complex is actually one of the important complexes in biology to produce energy. This is known as BC1 complex, which I'm going to talk about later. So what kind of energy are we talking about? Of course, everybody understands what energy is, but eventually what biological systems needs to do is to produce work. And work is not measured simply by energy, it's measured by what is known as Gibbs energy. So this delta G will be the change in the Gibbs, or it is also known as the free energy. Free energy is the energy free to produce work, in contrast to bound energy, which you cannot use for work. So that's why this Gibbs energy is so important for us to measure the deliverable energy, the energy which can be used for something useful. And so there are two components here. The first one is, of course, the energy. So this delta E means the change in the energy, and if you want to produce positive work, so this must be positive. So it's easy to see that the change in the energy must be negative, so which means essentially you have to reduce your energy to produce work, which is kind of easy to understand. If you want to do work in normal life, you basically have to reduce your energy. But there is another term which is not so easy to understand, and here we have T standing for temperature, and the next term is the change in the entropy. And again, if you see that from this simple equation, in order to get large positive work here, you have to have negative change in the energy and positive change in the entropy. So what does it mean? What does it mean to have positive change in the entropy? We could not understand that until the time when Boltzmann actually produced his definition of the entropy, and according to him, the entropy can be used to measure the number of configurations available to the system. So this W stands for the number of configurations. We can also call this W as the measure of disorder. If you have more configurations, it means you have more disorder in the system. And so the basic idea here is that if you go from one state to another, and so you have some number of states in the initial state, which will be W1, and you go to next step, which will be described by the number of state W2, so this number of states should be greater. And so that means that the entropy will increase and delta S will be positive in this process. If you think about that, it also means that the amount of disorder will increase every time you do some biological work. And that created the confusion that eventually, as a result of biology doing what it does, we should come to what was called entropy winter. Essentially, we are getting into a complete disordered world in which nothing can ever exist. So the question why it does not happen? We of course see in biology around us quite remarkable structures which reproduce themselves, and it means they do not go to this entropy winter predicted by Boltzmann. Schrödinger puts up this question in 1945. Essentially, the question was, how come that we still can exist and biology can reproduce itself, even though, according to basic thermodynamics, the ultimate result should be entropy winter. And so his idea was that biology somehow has internal mechanisms to compensate for this increased disorder, and he called it that biology feeds on negative entropy. And so this negative entropy became a very controversial subject, but later on, Brillouin suggested that this negative entropy is also can be understood as the information content in a living system. So essentially, the living system is capable of obtaining information from the outside world, store it, and in that way to keep itself ordered. Still, it does not explain how does it happen. So we still do not understand from this picture what is that negative entropy. So somehow it should come to a living organism, but where does it come from? So it became possible to understand the origin of biological energy when Peter Mitchell in 1961 formulated his chemiosmosis hypothesis, which proved to be correct, and it states that all biology's energy comes from gradient of concentration on the membrane of the cell. So here we have the membrane. Here is the inside of the cell, and this is outside of the cell. And his idea was that the concentration of the protons, which are just simple particles with positive charge, is higher on the outside compared to the inside. And so somehow this this difference in concentrations across the membrane allows you to keep the energy within the cell. So if you again look at the balance of energy here, here we have the membrane separating the cell into internal part and in the external part, and we have two things which actually define what is known as the chemical potential. So the chemical potential is the Gibbs energy per particle, and in this case this is per particle of the proton. So this is the change in the Gibbs energy that protons carry across the membrane. And it has two components. One comes from the difference in the electrostatic potential. This delta phi is the difference of electrostatic potential outside and inside of the membrane. And if you multiply it by the charge of the electrons and the charge of the ions involved, you typically get some number close to 150 millivolts. But again, there is another component. Remember this temperature which came to our Gibbs energy at the beginning. And this temperature multiplies now the next term, and this is the entropy. So this part is the same negative entropy Schrödinger was talking about. So again, the picture here is more complex than originally suggested by Schrödinger. We have both energy component here, so this is the energy part, and we have the entropy component here, but still there is a negative entropy which living systems are capable to store, but now the storage happens in terms of the potential across the membrane. So how this gradient of protons is created? So remember, we need to keep more protons outside the membrane compared to inside. So here I have more protons outside. That means the potential is positive outside of the membrane, and it's less positive inside. So this is my cartoon version of a cell, and this would be the membrane surrounding my cell. And so I need to create more protons outside compared to the inside. And the way nature does it, it sends electrons from outside to inside. So every time electrons comes across the membrane, it travels inside the so-called charge separation complex. So this complex essentially allows those electrons to move across the membrane. And so now this electron comes to inside of the cell, and here on the inside it can combine with the proton, and it means you eliminate your positive charge inside, and you create more charges outside. So now, how those protons outside of the membranes are used to produce energy, and for that we have a special complex in the cell which is known as ATP synthase. This is the complex which makes molecules which store energy, and those molecules are called ATPs. In order to produce those molecules and store energy in the form of chemical bonds, this is essentially where this energy goes. you need to send these protons from outside the membrane again inside the membrane. In the process of this flow, we create those ADP molecules. Essentially, we have a quite significant flow of electrons across the membrane. You need something like between 8 and 9 electrons to send across the membrane here to produce just one ATP molecule, and you have to have lots of them. Our typical body has about 250 milligrams of ATP, and 25 kilograms of ATP are produced daily. It means each of those ADP molecules inside the body is being recycled about 100 times in 24 hours. So now the question is where those electrons actually come from. And there are two ways to supply electrons. Plants and photosynthetic bacteria found ways to find electrons from most abundant substance on this planet, and that is water. If you take electrons from water, you produce oxygen, and that's why plants and bacteria actually created our oxygen atmosphere. And as a result, you get electrons from here, and then you can use these electrons to send them across the membrane. And of course, animals find electrons in a more straightforward way. They just use organic molecules from food to split them off and get the electrons for the energy. But most remarkable from all this observation is that all the complexity of the biology that we know of essentially comes down to two subatomic particles of physics. So electrons are small particles found by Thomson in 1897, carry the negative charge from outside the membrane to inside the membrane. So they essentially are carriers of Schrödinger's negative entropy to the cell. And once they're there, the energy is stored in terms of the gradient of protons concentration across the membrane. So the storage of this negative entropy is happening as the gradient of the concentration, meaning that the number of protons outside of the membrane is greater than number of protons inside of the membrane. Still, how those electrons move across the membrane? So the typical picture can be represented in the following way. We have a membrane separating the cell into two parts, and there is some energy input which can happen in two ways, either from photon of light, which is what is used in photosynthetic bacteria and plants, or it comes from food, which what animals do. And then after that, those electrons move across the membranes between molecules which are known as cofactors, which are the molecules which can take electrons and allow it to move on. So essentially, the electrons hop from one cofactor to another and finally go to the catalytic side on the inner side of the membrane where the protons are being consumed. Again, this movement of electrons across the membrane has the goal is to absorb those protons inside the membrane. And so in case of animals, the reaction is actually again very simple. We need to take oxygen in. That's why we need oxygen. And we combine it with protons and electrons to make water. So bacteria split water to get electrons, and we actually produce water from the electrons taken from food. Again, the general basic scheme of energy balance in all life turns out to be very general and very simple. So you need to create quite complex structures to move those electrons across the membrane, and this is the picture of what is known as the reaction center of biology, which is present in the membranes of photosynthetic bacteria. And this is a beautiful structure made of several proteins, so they are colored differently here. but the internal structure is quite remarkable. So these are cross-membrane proteins, and inside, shown by blue, you can see those cofactors, and as they light up, you can see electrons moving across the membrane. So this is kind of a schematic representation of what happens in such processes. This part of the complex, known as a primary pair, absorbs the photon. So here comes the photon with the energy h nu, and remember that energy is about 1.5 electron volt. That's the input energy for the entire process. And then the electron moves across the cofactors to the other side of the membrane to create the change in the Gibbs energy. Still, the question is what makes that electron move. And that question was answered by DeWalt and Chance in 1966. So what they looked at the rate of electron moving across the membrane, and here's the plot that they made from the experiment. So here you see the rate in the inverse seconds, and on this axis you can see inverse temperature, which means if as you cool, you go and in that direction. So here you have hot, and here you have cold. Okay. So you can see that at high temperatures, your rate of moving electrons is actually quite high. And then it drops dramatically as you lower the temperature. And it's at some point, it just stops changing and it levels off. So the behavior at the beginning at high temperature can be described by what is known as Arrhenius law, which tells you that the rate constant, K, is proportional to the exponent with the negative activation energy divided by temperature. So that part describes the initial part of the plot where the rate drops down with cooling. But then the lowest part of this plot describes essentially constant rate, and that corresponds to what is known as quantum mechanical tunneling. So quantum mechanical tunneling happens because electrons can move under the barrier without temperature being involved. But this plot essentially tells you that what we know, that life stops at low temperatures. And it does not require that low temperature to actually decrease this rate of moving electrons by a very significant number. You can see here, this is a so-called logarithmic plot, which means that the decrease here in the rate is about 8,000. So by the time you cool down to somewhere around this temperature, there will be no energy transfer present in the biological system. There will be no life. So why do we need temperature at when the rate constant is high? Again, you need to look at basic physics to understand what is happening, and you need to look at the phenomenon of electron tunneling. So this is a textbook example of electron tunneling under the barrier, and it starts from some energy level here, and it ends up at a different energy level on the particle known as acceptor. So here we have the donor and the electron tunnel under the barrier. So this square well describes the barrier for the electron to tunnel. The problem is that for electron to tunnel, you need to conserve energy. So in this situation, electron can do it because there is no energy difference delta E between two levels. But it's very hard to maintain delta E equal to 0 for different molecules. And what we usually see is something like that. And if you have two molecules in the gas phase with different energy levels, the electron will never tunnel. So why does it happen in real systems, in biological systems? In biological systems, we of course have high temperature, and what this temperature does for you, it creates thermal fluctuations. So the thermal fluctuations move this energy level back and forth around the average values, and it means at some point you create the conditions for resonance. It means electron can still tunnel even though on average there is a gap for tunneling. Which immediately tells you that you do need temperature to move electrons around, because because temperature creates fluctuations, and those fluctuations is what allows electron to move across the membrane by a sequence of tunneling events. So it is easy to see that fluctuations are useful for the purpose of delivering energy, and that's indeed what is happening in standard conditions. So this is the same reaction center of bacterial photosynthesis, but now placed in the computer, where all parts of the center are allowed to move. And you can see there are plenty of motions of the membrane here, and the red balls moving around here are phosphates, and this motion is of course transferred to the motions of the complex and to the cofactors shown here in green. So this is actually where the action happens. So electron hops between these green cofactors affected by the motions of the entire system. Of course, there are plenty of water molecules around here which you even do not see. So those blue wiggling regions are a big number of water molecules which move on much faster time scales compared to the motions of the membrane and the protein. So we are starting actually to understand the basic principles of biological machines. You need noise to overcome activation barrier. It means you need noise to provide conditions for electrons to tunnel between the cofactors. So being on the molecular size and being noisy in the thermal environment is actually quite important for energy to be delivered. At the same time, if you have too much noise, you have absolutely no control over your device. You don't want to lose control, and that's why biological machines cannot be too small. So they need to be small enough to use molecular scale noise, but they need to be large enough to allow repeated action and to be robust. So the scale of proteins is actually optimal for performing these two purposes. They can still feel the noise from thermal motion, but they are large enough in the size of hundreds and thousands of angstroms to be able to provide repeating action and provide some robustness to the entire operation. So now, what about our entropy winter? Remember Boltzmann's idea that eventually, as nature does its work, it should go to the state of complete disorder. So this is kind of a new topic and still being highly discussed in the field, and I just give you some idea what are the resolutions of this problem which are being discussed. So the problem is the following. You have some initial state which is called here W1, and you go to state W2. So this area essentially shows you the number of configurations in this initial state which would describe for you the entropy. And if you have the number of configuration in the initial state greater than in the final state, so then the second law of thermodynamics tells you that this transition is prohibited and cannot happen. So you cannot go from less ordered state to more ordered state. And of course, this is exactly what you want to do if you build a protein or you build the entire biological system, because you need to put some order into the system. So the protein molecule is highly ordered, and that needs to be built in when you synthesize the protein. How we are overcome this difficulty? The current idea is that biology uses information to decrease the entropy content. So essentially, this size of this region, the number of configuration, tells you about the extent of our ignorance about the system. So if you decrease the ignorance by providing more information about the system, it means you decrease the number of possible configurations and you decrease the entropy. So there is a trade-off between entropy and information. But once you provided information to your initial state here, you decrease the configuration space here, and it means you can now go from a smaller region to a larger region, and that transition is not prohibited by the second law of thermodynamics. But if you think about that, eventually what has happened in this process is that you moved from a larger region to a smaller region, which means you move to the state of smaller entropy and more order. So somehow biology knows how to use information to move to the state of higher order. We still do not understand it really well, but this is where the understanding of this problem is going now. Does it mean we are going to break the second law of thermodynamics? Of course, no. The point here is that all enzymes, all proteins, all energy complexes of biology act in cyclic action. So there is a cycle which returns the system to the same state. So this complex of biology works very hard to move those electrons across the membrane and provide your ordered state of stored energy, but then eventually it has to come back to the initial state, and at that point all the information content is released as heat to the environment. So the situation here can be described by this Greek character Sisyphus, who according to the legend was moving his rock up the hill until the point he would reach the top of the hill, and then the rock would roll back, and so the whole cycle would begin again. So the enzymes work in a similar fashion. They increase the amount of information available in the system by moving different parts of the system, but eventually they return back to the original state, and at that point the entropy is being balanced again according to the second law of thermodynamics. How many times those cycles can be repeated, of course, will define how long the organism will live. And of course, things happen, and sometimes those complexes fail. The reason is that these are still molecules, and the processes not always go to the intended direction. So as an example, I show you again the same BC1 complex of mitochondria in animals. And this is a large system in which electrons again move between those yellow cofactors, but those are just molecules. And if you zoom in, this is one of those molecules which is known as quinol, which is transferring electrons, but electrons does not necessarily go to where it is supposed to go. Like in this case, it's supposed to go to this cluster, but instead it can go somewhere else. And so for this complex, electron can go to oxygen, creating superoxide, and superoxide is highly damaging chemical substance which can damage components of your cell. So what happens in these cases is that these reactive oxygen species are created, which are potentially damaging for the cell and need to be eliminated. So antioxidants, which is a popular food supplement, has the role of removing such superoxide species from the organism. So I'm coming to the end of this presentation, and what I've shown to you is kind of the progress that we've made since about 1945, when Schrödinger put his famous question, "What is life?" And at that time, we knew really little about the building blocks of living organisms and how the energy is being provided to life. But the main question at that time, whether this complexity of life can be accounted for by physics and chemistry, essentially whether physical or chemical laws are behind of what biological organisms essentially accomplish. And the history of our development has shown that this is indeed true. So as I showed to you, two subatomic particles, proton and electron, is essentially responsible for the entire complexity of life and for the entire storage of energy in living organisms. So there are some fundamental physical laws like tunneling of electrons across under the barrier across the membrane, which account for the ability of organism to transfer energy. There are still big problems and questions which we could not understand so far, and one of them was also posed by Schrödinger, and this is the problem of consciousness. And so there are still many problems on the way to understand this basic question: what is life?